I will be presenting my poster titled Change in the Structure of Microbial Communities of the rhizosphere of peruvian fruit trees, Annona, cherimola, and Pouteria lucuma cross deep soil using PacBio HiFi sequencing. The soil microbiome is essential for ecosystem functioning as it influences diverse processes such as soil fertility, plant growth, nutrient turnover, and stress tolerance. However, much of our knowledge about the soil microbiome has focused on the surface layer, which has led to an underestimation of the role of the deeper layers, known as subsoil. Some of the factors contributing to the decrease in diversity at different tips are nutrient availability, gas and moisture levels, physical structure of the soil, and the competition and stress faced by microorganisms under these conditions. This research evaluated the microbiological structure of soil at three depths, 3, 12, and 30 centimeters, from two commercially important fruit trees in the South America region, lucuma and custard. About the methods. Rhizosphere soil samples were collected by shaking the 3 centimeters, 12 centimeters, and 30 centimeters roots of each plant. Then a soil analysis was performed, and finally DNA extraction, 16S RNA section, amplification, and sequencing were performed, and the 16S RNA sequence was obtained using the PacBio platform, which has the advantage of providing more precise information at the species level. Then all analyses and graphs were generated by our packages. The analysis, our results and analysis, we can see in this part. So in all samples, the relative abundance of the bacteria, phyla, Proteobacteria, Acidobacteria, Plantaceae, Actinobacteria, Firmicutes, and Gemmatimuriatis were the most abundant. The analysis of alpha diversity was determined by Shannon Inc. The results indicate that there are changes related to soil depth, since the highest indices were found in the samples near to the surface, 3 centimeters, and the diversity was reducing up to 30 centimeters. The beta diversity results show the rearrangement of the point of the bright curve dissimilarity matrix. It is observed that the lucuma and cherimola and custard apple samples have a clear separation in the principal coordinated plots, and the replicates of the samples are forming distinct clusters. Permanent analysis was carried out and determined that there are changes in bacterial diversity and composition by plant type, but not by soil depth, and there are differences when two factors, depth and plant type, are analyzed. In addition, the number of photos shared between the two plants evaluated was analyzed, and it was found that they share 1,157 photos in total. Specifically, 1,135 photos are exclusive to the lucuma plant, while 117 photos are exclusive to the custard apple plant. The same analysis was performed for each plant, considering the different soil depths. The results could suggest that possible specific soil characteristics at different depths or roots exudates of this tree may influence the presence and abundance of certain odors. The decrease in the amount of odors identified at greater depths could indicate that the soil conditions in these layers negatively affect the presence of certain microbial curves. The results of the soil analysis reveal the presence of certain factors in the liquid molar soil, such as heavy metals, which could be associated with a decrease in microbial diversity. This is supported by the results of Pearson correlation analysis, which shows a negative relationship between the presence of heavy metals and microbial diversity index. The results of the DC2 analysis indicate that the changes in the number of bacterial phyla in the bacterial community of the 30 centimeters and 3 centimeters lucuma soil. The result shows that there is a significant presence of 2 bacterial phyla in the 30 centimeters soil. The species identified as Telecola spinifila and Theobacter subterraneus. This last species is characterized by growing in anaerobic conditions and has the ability to oxidize sulfur compounds. In conclusion, in soil diversity, different types play a crucial role in ecosystem and in the cultivation of important plants. The studies have shown that microbial compositions vary significantly along the soil profile, and factors such as nutrient availability, heavy metals concentration, carbon content, and other soil properties influence this diversity. Understanding microbial composition at different soil types can help optimize agricultural practices such as fertilization and associated crop selection to promote soil health and improve the production of this plant. I would like to thank the Peruvian Society of Bioinformatics and RSG Peru for making my participation in this event possible, their sponsorship and continuous help.